to model and develop models of human disease, to begin screening for compounds, and understanding the mechanisms of these diseases are here. So now that I've said that, I want to go back and say, what about the brain? And what about the mature brain? What are we left with there? Are we going to always have to rely on reverting cells back into an embryonic state to repair or model human disease? And this is a very famous scientist that we all, in our early career, studied, is Ramon Cajal. He got the Nobel Prize for his discoveries, and he made this statement that has stuck with us, which says that once development has ended, the font of growth and regeneration of axons and dendrites, which are the processes of neurons, the connections, have dried up irrevocably. In the adult centers of the brain, nerve paths are something fixed, immutable. Everything may die, nothing may be regenerated. And I learned this as a kid, that this, once the brain has reached a certain stage of maturation, it's fixed, and it's basically a computer. It calculates and responds to the environment, and as we age, it basically deteriorates. Look, and this is not that hard to, even as a biologist, it's not that hard to think that this is true. The brain, human brain, is made up of something like 100 billion neurons, 100 billion cells, 100 billion neurons, and there are 100 trillion connections between those connections. These numbers, of course, are hard to fathom, except for the fact that in this economic recession, these numbers are actually taking on a new meaning for some of us. These cells are large, they have all these connections, so how could it be that a neuron, and how could a neuron with all these connections, how could it actually divide and make more neurons? So that dogma that the brain had no capacity for regeneration or making new brain cells stuck with us as scientists for a long time. But it turns out, and this is just a cartoon of what a neuron looks like, with the cell body, and up there are the dendrites, which are like the antenna receiving connections, and this long blue process is how it communicates with other cells. So imagining that cell could actually divide and make two cells, and with all the appropriate connections so it wouldn't screw things up, is really hard to fathom. So, but it turns out that the dogma is laid in pretty good grounds. And it really holds true. Brain cells like that do not divide. But the surprise is the brain has stem cells in it. The brain contains immature cells that can self-renew and give rise to new brain cells. If this occurred everywhere in the brain, this would be dangerous, my guess is, and that it would be hard even for a lay audience to consider. If every cell in the brain could divide and was dividing and replicating and making new connections, it would be one would certainly worry about tumors much more than we do now, but there would be a difficult way to conceive of any consistency. How do we retain memories? How do we retain any concept of ourself if the brain, which is the organ of our behavior, is changing all the time? So it turns out that it's not occurring everywhere in the brain. It occurs in only a very few areas of the brain where these stem cells exist, but they're in very critical areas. And one of the areas where it occurs in a structure called the hippocampus. And a hippocampus is a word that means seahorse, and it was called seahorse because of its shape. It has this beautiful, I say it's beautiful because I've been studying it for 40 years, but it's a wonderful structure. And within this structure of the brain, which is important for making new memories. So if you remember anything from this, the way it happens is that you're seeing me and you're hearing me, you're having some emotional response to it. While you're hearing and seeing me, you're trying to relate to what I'm saying in some way and relate it to something else so that you can try to understand some part of it. We always try to do that sort of thing. You're being affected a little bit by the person next to you based on their smell and some other sensory things that are happening. You're worried about how long I'm going to go on and all these sorts of things. All of those factors come in at one time to contribute to what it is that you will ultimately remember. And the structure of the brain that's involved in putting all those elements together is this structure called the hippocampus. So memories are formed here, but they're not stored. They're stored in other areas, but they're aggregated into the single memory. So that when you try next week to remember this, you will not remember just my face or some physical feature of me, but when you recall that physical feature or you recall my voice, then my image will occur, and the other feature, the person next to you and all the other stresses, all of that comes back because the memory has now been encoded as a unit. And this is the structure that does that. And interestingly, that's the structure where these stem cells are, where new cells are being born all the time. So this is sort of a little cartoon that probably won't work. Oh yeah, here we go. So this is the structure where it happens, and there's a stem cell, and the stem cell, I haven't tried this in public yet, so see if it crashes on me. So they divide, and this is not just one cell, but there are many cells, but they're all at this one level. We make more cells are being born than we actually use. Some cells will die, and the reason they die is because we don't use them. We don't activate them. But some of them will survive, and they send out their dendritic branches, and they integrate into the circuitry. So that's the process by which neurogenesis occurs throughout life, albeit it's highly regulated. And here's just some images I'll show you of what by using these techniques that we have for visualizing these cells. All the green cells, it's not showing up so clearly, but all the green cells with their processes correspond to new cells that were born over a period of about 3 hours in the brain. So in this structure, there's a fair number of cells being born at any one particular time. And this just shows an important fact, and that is, in the adult brain, the process by which these new cells are born in the brain is extended over a period of months, because you can imagine these are very complicated cells, and it takes time for them to extend their processes and make connections within the brain structure. So neurogenesis is not a single event. The birth of new neurons is not a single event. And what you do during the period of time that these are being born in your brain will affect their survival and their ability to interact. And we know now that all mammals in this structure of the brain have this very same process, from mouse to human. And interestingly, all of these animals have this same structure, and this same structure, this dentate, this hippocampus, is involved in this memory. Of course, all these mammals need to remember, to make new memories, which is what the hippocampus is doing. Quite remarkably, we can take these cells out of the brain. You can do a biopsy, you can do an autopsy, more likely even. Harvest the cells, and we've developed techniques for spinning them down, and you can isolate them as individual cells in a dish, and these are from the adult brain, and that green cell there is, in fact, the stem cell been characterized as an adult multipotent, not pluripotent, so it can only give rise to other cell types, but it can divide in a dish. And we can find conditions which can optimize the ability of these cells to retain their stem cell properties. So these cells will continue to divide. This is symmetric, that means they're giving rise to themselves indefinitely. And they'll continue, and they're down at the bottom on the left is a time course of how this is happening. And they have an intrinsic clock, so they'll divide about every 24 hours, and they just split. And what happens is that the cells will continue to divide until the culture dish gets so dense that they start bumping into each other, and they start signaling to each other, and then they'll differentiate and become different cells, mature brain cells. But at this point, they're stem cells. But this is what happens to them if we put them under the appropriate conditions. they can develop branches and connections just like they do normally in vivo. And we and my colleagues now have figured out ways of taking these adult stem cells in culture and either propagating them indefinitely or finding ways to induce them to differentiate. And part of the reason for doing this is that we want to figure out ways to activate these cells endogenously so that they can survive better, make faster neurons and better neurons. So what this means then is that in addition to the concept of transplantation or drug therapy, a new field of investigation, which is called self-repair, is emerging. And in this case, what you're doing is targeting those cells, the remaining stem cell population, and targeting them to survive better and to integrate better. And we're also trying to figure out ways to not get them to go just in this area, the hippocampus, but can we make them migrate to other areas where cell loss occurs? And many labs around the world now are working on this, and even several labs here in Melbourne as well are working on this area. So in addition to this cartoon I showed you of embryonic stem cells making neurons that can transplant, we can get biopsy material from humans, and we may even be able to activate the cells endogenously. So the last piece of this is that not only do the cells occur under normal conditions, but they're highly regulated. That means that as we age, they decrease. As we, under disease conditions, they decrease. However, under enriching conditions, if we enrich our environments, have more experience, different experiences like you so brilliantly are doing tonight, you're actually affecting the survival of these cells in your brain. That's amazing. And physical exercise. If you walk home tonight or whatever, you can do that as well. So here's some experimental differences that were really quite remarkable when they occurred, when they were developed. So normally, over the left, little mice are housed in these small cages. If you took them and put them into a more complex environment just for 1 month as adults, the control is above, and those black dots all correspond to new neurons that were born in that structure. You can see that in column A, D, and G, that just by a month of either physical activity or enrichment, cognitive enrichment, many more cells were grown. And here's actually the numbers for it, because it is really quite remarkable. In this structure, there are about 270,000 neurons, right? The animals that were exposed to a cognitively enriched environment for 1 month ended up having 50,000 more neurons surviving 1 month later, which was 15% of the whole volume in the structure. So structural, physical increases in activity can occur by this. So here's an experiment. This is the equivalent, right? You know. And we have these little treadmills. We actually use running wheels for the mice as well. The distance, the active distance moved by an animal is linearly correlated with the birth of new neurons. So